The human body is 45 to 75% water. So let's look at the factors that determine where you are in that range. The first factor is age. And in age, the younger you are, the higher your water percentage. In fact, babies win the prize for the most water. Babies are at 75% water. Think about that. A newborn baby is 3/4 water. Elderly win the prize for the lowest. An elderly person is at that low end, 45% water. Another factor that goes in is body composition. And here we're looking at lean and obese. So if you're obese, you have a lot more fat or adipose. And remember, fat is hydrophobic. So fat and water repel each other. So the more fat you have, the less water you have, because all that fat is repelling water. So somebody who's lean has a higher water percentage because they don't have the adipose to repel the water. And then the last one is sex. And in this case, men have more water. If you take a man and a woman that are the same age and either both lean or both obese so that you have an apples to apples comparison there, the man will have more water. The reason for this is that women have estrogen. Estrogen causes the growth of adipose. When a girl goes through puberty, she gets more adipose on her body. So estrogen stimulates adipose growth. And as we already talked about, adipose repels water. Men have testosterone. And testosterone stimulates muscle growth. And muscle is very high in water. So the more muscle you have, the higher your water percentage. So now we're going to look at how all this fluid, along with the electrolytes that are in it, is distributed throughout your body. So in the book, you read about the rule of thirds. So that summarizes the distribution of the water in your body. First, we're going to divide into two main categories. You're going to have intracellular, inside your cells. So ICF is intracellular fluid. This is what is inside your cells. This is also called cytosol. Cytosol is the water inside your cells. This is 2/3 of all the water in your body. So these rectangles are drawn to scale. So from here to here, that is 2/3 of the entire rectangle. Then you have extracellular fluid, ECF. So ECF stands for extracellular fluid. This is all of the fluid outside your cells. This is 1/3. So from here to here is 1/3 of this rectangle. So of all the water in your body, 2/3 of it is inside your cells, 1/3 of it is outside your cells. ICF inside your cells, ECF outside your cells. Then this can be divided into two more groups. So ECF is divided up into two more groups. You have interstitial fluid. This is all the fluid that's just between your cells. And you have your blood. If we look at how ECF is divided up between interstitial and blood, so keep in mind here, interstitial and blood are the two parts of ECF. Interstitial is 2/3 of ECF. So it's 2/3 of extracellular. So interstitial, which is also called tissue fluid, this is the water between your cells, is 2/3 of that 1/3. Well, if you multiply your fractions, you just take 2 times 1 is 2, 3 times 3 is 9. That is 2/9 of all the water in your body is your interstitial fluid. Blood is 1/3 of extracellular fluid. So all the water in your body, 1/3 of it is extracellular, and of that 1/3 that's extracellular, 1/3 is in your blood. So blood is 1/3 of 1/3. 1 times 1 is 1, 3 times 3 is 9. So 1/9 of all the water in your body is in your blood. So that's the distribution of water. Now let's also look at electrolytes. Remember, think back to chapter 2. You first learned about electrolytes in chapter 2. They're in your food, right? Calcium, sodium, potassium. Those are all components of food. So you get your electrolytes by eating food. And remember, electrolytes dissolve and dissociate in water to produce your ions. So now we're going to look at these. Inside your cell is where you have the majority of your potassium, your phosphate, magnesium, and you have proteins inside your cell. Now remember, you've learned about sodium-potassium pump. So we have a sodium-potassium pump here. That's why potassium is inside. The pump pumps it in. And remember, it's a sodium-potassium pump. So if it's pumping potassium in, it's also pumping sodium out. So here in your extracellular fluid, you have sodium. You also have calcium, bicarbonate, and chloride. Those are the main components of extracellular fluid. Blood has sodium, calcium, bicarbonate, chloride, and proteins. So now let's look at some information comparing these areas. Inside the cell, the interstitial fluid, and the blood. So which two fluids have protein and why? Well, you can see that your intracellular fluid has protein and your blood has protein. So ICF, the reason there's proteins inside your cells is because cells have ribosomes that make proteins. And the reason there's protein in your blood is because your liver, remember chapter 18, your liver makes plasma proteins and puts them in your blood. Also, chapter 22, your liver makes complement, part of the immune system, and puts those proteins in your blood. So your blood gets proteins from your liver. It gets plasma proteins and complement from your liver. Okay, so the one with little or no protein is interstitial. The reason for this is it has no source of protein, And the proteins that are in the cells and in the blood can't cross the cell membranes and the blood vessel walls to get to the interstitial. So the proteins in cells can't fit through the cell membrane to get out. And the proteins in blood can't fit through the capillaries. Proteins are fairly big molecules. So you have your blood is gaining and losing things based on what you eat and drink, and your kidneys filtering your blood. Your cells are making things, they're breaking down molecules, they're making molecules, so they've got stuff going on in there. The interstitial fluid is just kind of this middleman between your cells and your blood, and it doesn't really have much control over what's happening to it. It's just getting whatever your cells and blood decide to give it, and it's losing whatever your cells and blood decide to take from it. Okay, the two that are the most similar are your interstitial and your blood. And the reason for this is because capillaries have holes. Remember, you've learned this. Capillaries have holes. You want exchange. You need stuff moving in and out of the blood at capillaries. And really, the only difference between the interstitial and the blood is the proteins. Everything else you notice is the same here. So all of this is the same. And the only reason that the proteins are a difference is because proteins are too big to fit through those holes. Okay, then if you look at sodium and potassium, notice they're kept separate. Potassium is inside the cells, sodium is outside, and that's because cells have sodium-potassium pumps. And those pumps are constantly moving sodium and potassium in opposite directions of each other. And just to point out, it's most of your potassium is inside here, and most of your sodium is outside. Not all. There is a little bit of sodium in cells. There is a little bit of potassium in extracellular fluid. So this is most, but not all. Okay, then if you look at calcium and phosphorus, those are also kept separate from each other. You have phosphorus inside, you have calcium outside. You purposely do this. You make sure you keep calcium out of your cells. So you also have calcium pumps here to keep the calcium out. You don't want calcium inside your cells. Well, think back to chapter 7 about bones. When calcium and phosphorus get together, they form hydroxyapatite, and that is what makes bones hard. Well, you only want your bones to be hard. You don't want all the other cells in your body to harden up. So they are kept separate to prevent the formation of hydroxyapatite all over your body. So you only want hydroxyapatite in your bones. Your bones are the only part that should be hard. You don't want your whole body hard like bones. Okay, so that takes care of the distribution of the fluid and the electrolytes. Now we're going to look at osmolarity. So this goes back to chapter 4. You learned about osmolarity in chapter 4. Remember you did the lab where you put a potato in salt water and we had the tube of molasses and the water came into the molasses. So that's what we're looking at here is osmolarity. This is basically how salty a solution is. And remember, salt, when you're talking about it in science, doesn't just mean table salt. So we're talking about solutes, things that attract water. So we're looking at the attraction of water. And remember back in chapter 4, you learned about isotonic, hypotonic, hypertonic, and how it's important to maintain that, because otherwise your cells would either shrivel up or explode because of the attraction of water in or out of them. So we're just first going to start off by looking at basic osmolarity. And I have beakers here set up, and we're going to have the top one is just going to be our standard for comparison. And I'm just going to use very simple numbers. So these do not represent the actual amounts of water and salt in your body. I'm just using simple numbers to make the point. We're going to say that here we have 10 water and 10 salt. That means we have a ratio of 1 to 1. 1 water for 1 salt. Now down here we're going to do different varieties of water and salt in the beakers and see how it compares to our standard. So we're going to start off, let's say we keep the water the same. We still have 10 waters, but we double the amount of salt. We have 20 salts. Well, now if you look at the ratio, you have 1 water, but you have 2 salts. So you made this saltier, right? If you add more salt, it gets saltier. Well, there's another way to make it saltier. We can also reduce the amount of water. So let's say we keep the salt steady at 10, but we cut the water in half to 5. Well, you still have that 1 to 2 ratio now. This is still saltier. In each case, whether you raise the salt or you reduce the water, your concentration of salt is higher now. You have saltier water. And this is increased osmolarity. And whenever you increase the osmolarity, you're going to attract more water. If it's saltier, it will attract more water. Okay, in the middle one here, we're going to keep a constant osmolarity. If we double the water and we also double the salt, nothing really changes as far as osmolarity. It's still a 1 to 1. If we cut both in half, if we only have 5 water and only 5 salt, again, it's still 1 to 1. We didn't change the osmolarity. So this is constant osmolarity. So if you change the water and salt together so that either they both increase or they both decrease, your osmolarity stays the same. Okay, now let's look at how we can reduce osmolarity. So we're going to make it less salty. Well, we can decrease the salt. If we keep the water the same, but we knock the salt down to 5, you now have 2 waters for every 1 salt. Or you can increase the water. So we can keep the salt steady at 10, but we can double the water. And again, you're going to have 2 waters for every 1 salt. So in both of these cases, we diluted the salt. We made it less salty. So this decreases the osmolarity. Anytime you reduce the saltiness, you decrease the osmolarity. And so this is now less attractive to water. So if you make something saltier, that increases the osmolarity and it is more attractive to water. If you make something less salty, you decrease the osmolarity and that is less attractive to water. Okay, so let's go through these questions. 2 ways to increase osmolarity. Well, you can increase osmolarity by increasing the salt, or by decreasing the water. Either of those actions will make it saltier. Two ways to decrease osmolarity. So now look over to the right. If you decrease the salt or you increase the water, you make it less salty. And osmolarity is not just the amount of salt, but it is the ratio of salt to water. Or you can think of it as the concentration of salt, right? If we start with a beaker with 10 water and 10 salt, and we add 10 salts to that, but we also add 10 waters, we didn't change the osmolarity, even though we added more salt. It's about the salt and water ratio, how they relate to each other. Okay, so now let's put that in context of cells. And again, this is going back to chapter 4. This is a review of chapter 4. We'll have 0.9 in each cell. That is the saltiness. A cell is 0.9% salt. So we're going to start off with, in the middle, we'll do isotonic. Okay, so remember, iso means equal. So this means we put a solution outside the cell that is also 0.9% salt. So this is a constant osmolarity. Neither one is any more attractive to water than the other, so water will stay put. And remember, this is what you want. Again, go back and review chapter 4. In your chapter 4 lab, this is where we covered this. You want water to just stay put. Let's suppose you have hypertonic. Remember, hyper means more. And remember, with these words, we're talking about the outside of the cell. That means the outside of the cell has more salt than the inside. So let's double it. Let's say 1.8. 1.8 is twice as much as 0.9. This means you have an increased osmolarity out here. And remember, if you increase the osmolarity, it is more attractive to water. So water will leave the cell. Because it wants to be with that higher osmolarity on the outside. And this causes crenation of the cell. So the cell will shrivel up. And this is bad. You don't want this. You'll die if this happens. And the example, if you remember back to chapter 4, this is if you drink ocean water. This is why if you're lost at sea, you can't drink the ocean water. Ocean water is 3.5% salt. That will make your blood very hypertonic. It'll draw the water out of your cells. Okay, let's look at the other end here. You could have hypotonic. So hypo means less. So let's say this is 0.45. 0.45 is half of 0.9. So if that is the case, you have a lower osmolarity out here in your extracellular fluid, your blood and your interstitial. That means the inside of the cell is more attractive to water. So water moves into the cell. And this will cause lysis. Your cell will explode. And this also causes cerebral edema. Causes the brain to swell. And remember, that can cause seizures and death. So if you relate this to our beakers, the way you would become hypertonic would be by adding more salt or reducing the amount of water. So like I said, with ocean water, it's 3% salt, 3.5% salt. That's adding salt. Or if you're not drinking so that you become dehydrated. So this would be if you add too much salt to your blood, or you fail to add water to your blood, you get dehydrated. Okay, this could also be like sweating a lot and not drinking to replace it. So it's true there is salt in sweat, but there's more water than salt. So when you're sweating, you have a net loss of water. And with the hypotonic, hypotonic, that would be the beakers over here on the right, where you are either you're diluting it by either adding too much water or you're not adding enough salt. So this would be like, for example, if you drink too much water. Remember in chapter 4, like the Wii for Your Wii, that contest the radio station had where you had to drink a bunch of water and then you won a Wii gaming system, and the woman who won died because she drank too much water and made her blood hypotonic. And this could also be from a very low sodium diet. Okay, so what happens if you drink too much plain water? Well, if you drink too much plain water, you will make your blood hypotonic. Because you will dilute the salt that you have in your water. You're adding water, but you're not adding salt with it. What happens if you sweat a lot or don't drink enough water? Well, in that case, you would make your blood hypertonic. Because remember, sweat is salty, but there's more water lost than salt lost. And IVs are 0.9% saline. Sometimes they're 5% dextrose. That's the other option, because that is what is isotonic. And this way your cells neither gain nor lose water, so they stay alive. Remember, if they gain water, they'll explode. If they lose water, they crenate.